All right, well, hello everyone. So what we're going to look at here is this process of cellular respiration. Okay, so cellular respiration, big overarching idea is we are using some nutrient source, and in this case we're going to talk about sugar, so glucose, and we're going to change that. We're going to convert it essentially into energy, right? So we're trying to make ATP here. So we're trying to make adenosine triphosphate, okay? So basic formula here for our cellular respiration, we're using glucose, so C6H12O6, oxygen, so O2, and then we are producing a couple of things. One, we're producing carbon dioxide, so CO2 as a waste product. We're producing water, H2O, and then kind of most importantly, what we're going for here is to produce energy, right? So we're making energy in the form of ATP, right? So we're going to talk about various stages or phases of cellular respiration. And for each of these phases, stages, what I want you to think of is, one is, where is it occurring? So where in the cell does this phase occur? What do you start with? What do you produce? And then is it aerobic or anaerobic? So meaning, does it require oxygen or does it not require oxygen? Aerobic meaning it requires oxygen, anaerobic meaning it doesn't. Okay. So this first phase, this first stage of cellular respiration is glycolysis. Okay, glycolysis. So if you look at the word glycolysis, glycolysis is split into two words, glyco and lysis. Glyco is referring to sugar, so glucose, and then lysis means to split or to break apart, right? So in this case, what we're doing is we are splitting sugar, right? We are taking this large 6-carbon molecule. So again, glucose is C6H12O6. So pay attention here. On this right side here, we are producing, or we have a 6-carbon molecule. And what we're going to do is, through a series of 10 steps, all facilitated by enzymes, we are going to take the 6-carbon glucose and we're going to break it into two 3-carbon molecules called pyruvate. Okay, called pyruvate. So again, what do we start with? We're starting with glucose. What are we producing? We're producing pyruvate. Pyruvate is a 3-carbon molecule. We're producing two of them. So one 3-carbon molecule, a second 3-carbon molecule. There's our 6 carbons. So we didn't lose any in the form of, say, carbon dioxide. This is occurring in our cytosol. Okay, so cytosol being the liquid aqueous component of our cell, right? So think outside of the nucleus, but inside the plasma membrane, right? That liquid, that's the cytosol. Okay. And then this is an anaerobic process. So meaning we don't require oxygen. Okay. This will occur if oxygen is present, but it just doesn't need oxygen. It's not dependent upon oxygen. Okay. So this glycolysis can be split into two phases, called the investment phase and the payoff phase. Simply put, the investment phase, think you are investing energy into the system. So in other words, we have to use ATP. Okay, we have to use energy and phosphorylate these various molecules here. Okay. An enzyme that phosphorylates or adds a phosphate group, that is called a kinase enzyme, K-I-N-A-S-E. The ending A-S-E, you know, it's an enzyme, but kinases are specifically enzymes that phosphorylate other molecules. So again, adding a phosphate. Okay, so we're using an ATP here, and we're using an ATP here. So we're down to ATP, right? So remember, this whole process that we're trying to do, we're trying to make ATP. Okay, we're trying to make ATP, but initially we have to use some ATP, right? So we add it here, so we go from glucose to glucose 6-phosphate. The phosphate is attached to this 6th carbon. We change that slightly into fructose 6-phosphate. This gets changed into glyceraldehyde 3-phosphate, or G3P. And again, in these two phases, we're using energy. So this is the investment phase. We're investing energy into the system, right? What then happens is this payoff phase. This is happening twice. So notice these 2 times indicators here. So what happens for one, whenever we go from this glyceraldehyde 3-phosphate, which is this 3-carbon molecule, once we go through it here, we're making 2 ATP, and we create these pyruvate. Remember, we make 2 of them. We go through this once, and we make 2 ATP, and we go through it again, and we make another 2 ATP. Okay. So what we end up with is we end up grossing. We make a gross total of 4 ATP, but because we used 2, we only net 2. So notice here, net gain of 2 ATP. We gross 4, but we only net 2. Okay, so be careful in terms of how you are, how you're thinking about this, in terms of whether you're talking about gross number of ATP, which would be 4, or net, which is subtracting our investment phase, the number of ATP we used in our investment phase. Okay. So we make our pyruvate, we make a net of 2 ATP, and then we also make these things called NADH. Okay, NADH. These are molecules that are essentially high energy storage molecules, and essentially for now, think of them as being like pre-ATP. Okay, so meaning they'll be used to make ATP, but they're not ATP directly. Okay, so they're not adenosine triphosphate in and of themselves, but they will be used to make adenosine triphosphate, right? Another thing you can think of is for every NADH, that is going to contribute to the production of about 3 ATP. So if we have 2 NADH molecules here, we've made, ultimately, this will contribute to about 6 ATP, and then we have 2 here. So we're at 8, but in terms of actual ATP, again, really only 2, right? A net of 2, okay? So another way you can picture this phase is with a little bit more detail. And so there's a lot of things going on here. And so this is just a slightly more in-depth picture of what's happening here, especially this bottom here. So notice here, we are using ATP right here. So we do this twice, so we're using 2. And then on this side over here, we are making 4. Okay, so this is happening twice. So 1 and 2, 3 and 4. So again, we net 2, right? We net 2. This down here is just the same thing as this up here, but just with more detail. Now, for this class, I'm not overly concerned if you know the names of the specific enzymes, but I do want to draw your attention to the name of some of them, especially those kinases, right? So look everywhere where you have a ATP shown, and you'll notice that the name of the enzyme ends in kinase. Okay, so here we have hexokinase. Here we have phosphofructokinase. Here we have phosphoglycerate kinase. Here we have pyruvate kinase. Right. So remember that kinase means an enzyme that is phosphorylating. Right. It's adding, it's removing, it's doing something with a phosphate group. Right. So if you look here at the very end, what do we end with? We end with a pyruvate molecule here, which is 3 carbons, and another pyruvate molecule here, which is also 3 carbons, right? Now, what happens to this pyruvate, right? What happens to this 3-carbon molecule? Well, that's completely dependent on whether or not we have oxygen present, okay? So if we have oxygen present, then that pyruvate is going to enter into essentially what is this kind of intermediate preparatory step called pyruvate oxidation. If we don't have oxygen present, so if we're still in anaerobic conditions, then that pyruvate will get converted into lactate, and this allows the process of glycolysis to continue even when you don't have oxygen, but that lactate will build up. It's transported to the liver, it can be converted, it'll be stored there, it can then get reconverted ultimately back into glucose, and then proceed through the step again, right? And that's a process called the Cori cycle, and we won't talk anymore about the Cori cycle, right? But just know, if we have oxygen, so this slide here, if we have oxygen, we're going to go from pyruvate into something called pyruvate oxidation. If we don't have it, then that will get converted into lactic acid and be sent to the liver. Okay. So this is where we're going to stop for this part. Okay, so for glycolysis, what we'll pick up with in the next part is looking at what happens to that pyruvate, assuming we have oxygen present. What's going to happen to that pyruvate after it is formed?